Let me share my screen. So yeah, is my screen visible?
No, sir. Yes, sir. Actually visible.
Now? Okay.
Yes, sir.
So welcome to this interaction session once again. So today we have just prepared a few slides regarding the queries we had got earlier. So I'll first take up the questions which we had got earlier. Like there was a question regarding the crystal habit. My voice is clear.
Yes, sir.
Yeah. So the crystal habit is nothing but it's the outer appearance, how the crystal appears, or its geometrical shape basically. So that we call as the crystal habit. Suppose there are a few crystals which we obtain in the form of needles. So that is one type of crystal habit, and there are few crystals that are obtained in the form of isodiametric. Isodiametric means the diameter of the crystals will be similar from all the sides. So these are isodiametric crystals, and there are cubes, there are blades, there are plates. So these we categorize, we call as the crystal habit. So what is the importance of this crystal habit? Because as we know the shape of the crystal, it uh, it causes different properties. Like suppose there is flowability. If you consider the property of flowability, we know that isodiametric or spherical shaped crystals, they have a very good flowability. And when we compare those to needles basically they exhibit poor powder flow property. So these are some of the aspects which the crystal habits will be interfering with, like also the compaction properties and the filterability, the dissolution rate, etc. So it's important to control the crystal habit of the compounds, because if we get different habits in different batches, then it will lead to different properties. Maybe the dissolution rate of this particular crystals will be different from the dissolution rate of another batch. Suppose I'll show you one example which we had got in our lab. So suppose there was ethyl hydrochloride which we crystallized. So when we used one particular solvent, we got rod shaped crystals. Okay, so these rod shaped crystals, they showed very poor powder flow properties. Wait, let me turn on my pointer. Yeah, so these crystals had very poor powder flow properties. And in other solvent we got plates. These had comparatively better flow properties. So we need to maintain the crystal habit constant in every batches. So that's the importance of crystal habits. And there was another example of our lab itself. We got three different crystal habits of the same compound. Like one was the needles, and a mixture of rods and plates, and then just plates. So all these three different crystal habits, they show different dissolution rate, and then they also show different flow properties, different degree of compressibility into tablet. So this all changes with the crystal habit. And there was another question regarding the aspect ratio. So as the longest side of the crystal, we'll consider that as the length. Okay, so divided by width. Suppose the length is like 500 micrometer and the width is like 50 micrometer. So 500 divided by 50, it will be 10. Aspect ratio of 10. Is the importance of this aspect ratio? We decide the crystal habit on the basis of the aspect ratio. Suppose like as you can see in this table, there are different types of crystal habit and they have a characteristic aspect ratio. Suppose acicular or the needle shaped, they will be having aspect ratio of usually more than 10, and similarly for fibrous, they will be having aspect ratio of more than 10. And if rods, the aspect ratio will be between 3 to 10 generally in general cases. And the plates 2 to 3, the blocks 1 to 1.5, spherical it should be basically 1, because all the sides the diameter will be the same, so there won't be any changes, it should be 1. So for more information on this particular topic, I suggest you to read this particular article by Ram and Dhunale et al., published in Crystal Engineering Communication. You will get a in-depth idea about the crystal habits and its effect on the downstream processability, such as the filterability or dissolution rate, and how can we control the crystal habit, what experimental procedure can be followed, etc. You can find in this particular publication. And also there was a question regarding the amorphous compounds, like which compound or which material has greater solubility among among the amorphous or the co-crystals or the crystalline substances. So yes, amorphous substances, they have a very higher solubility compared to other two, like the co-crystals and one second. Yeah, sure. I'll share the reference publication link. Towards the end of the session, I'll share it here. So yeah, as I was talking about the amorphous substances, they have a very random arrangement of molecules. Because of that, the entropy of the molecule, the entropy of the molecule increases. So as we know, entropy is the degree of randomness. It just says how much energy the molecule basically holds or the compound really has. So higher the entropy, higher the energy. So due to their higher energy, they have a very good solubility profile. There's no doubt in that. But also due to this higher energy, they tend to become unstable because of this high energy. So the aspect which is making it one second, I got another message. Screen is not visible. Okay. Am I visible now? If my screen is visible? So yeah. So I was telling about the energy aspect. So the thing that makes it to be very highly soluble in water, the same thing will also cause it to be very unstable. So because of its instability or high energy, it will tend to recrystallize. So in the meantime, suppose just imagine you prepared a amorphous material. At the time when you're releasing the batch, it is having a very good solubility. But over time, it tends to recrystallize. That is, it will undergo rearrangement because it has very high energy. That has to be again, it has to get back to its normal form, because any molecule which has higher energy, it will tend to lose its energy to the environment, and it wants to become stable. So in the due course, maybe after 6 months, it turned into crystalline compound. Now, as we know that the crystalline compound, it has low solubility. So when at the time of the batch release, the solubility was very good, and after 6 months, the solubility reduced. So this is not acceptable by the regulatory standards. So that means the batch is failing. It is failing the stability test. So that's one of the disadvantage of amorphous material. Like there has to be a lot of care that has to be taken while storing these kind of materials to avoid the recrystallization. We need to reduce the temperature, maybe to reduce the rate of recrystallization, or we need to use some polymeric additives, or we need to use specialized storage containers or something which does not permit the moisture entry, because the moisture can enhance the crystallization rate. So these are the few disadvantages of amorphous materials. And yeah, mostly around 90%, or I can say 80 to 90% of the drugs in market are currently in crystalline form, because they have a very good stability and reproducibility, as well as good manufacturability. But in cases where the solubility is a must, and we can take the risk of the stability, and we have like figured out a way to stabilize these amorphous compounds, then we can surely go for amorphous. But yeah, that is very again costly, and it needs specialized procedures to be followed. So yes, and we can prepare these kind of, also these amorphous materials, they are susceptible for degradation, because they have very high energy. High energy basically relates to reactivity. So they have very high reactivity, and they also tend to degrade faster compared to their crystalline counterparts. And also they show a little bit of hygroscopic behavior. They tend to take up moisture from the environment during storage. So this makes their handling and all difficult, and then we need to bring in some engineering controls to, yeah, to basically stabilize these compounds. And they can be prepared by various methods, like they are prepared from crystalline materials itself. Like we can quench cool after melting, or the hot melt extrusion process, or by spray drying, co-precipitation method. Novel ones are the electrospraying method. Milling can also be used to induce amorphization in compounds. And there's also vapor condensation, which is not widely followed, but yeah, these were the primitive methods that were developed earlier. Most widely followed are hot melt extrusion, spray drying, precipitation, and milling process. And there was also a question regarding co-crystals, like how they, what are their advantages, how they are formed basically. So co-crystals are nothing but these are crystalline materials where we use more than one component. Like there's API, and also there's another component called as coformer. These two components are linked to each other, or they form a kind of there is no covalent bond interaction here. They form hydrogen bond or van der Waals interaction, or there can be pi interactions. These kind of weak interactions are involved between API and coformer. So they further crystallize in the same crystal lattice. There is in a definite stoichiometric ratio. Stoichiometric ratio is, suppose if you consider the molar ratio, if you consider one molecule of API, there might be one molecule of coformer in some particular co-crystals, or like like we can see in the palmatine sulfosalicylate co-crystals. For every molecule of palmatine, there is one molecule of sulfosalicylate, which is a coformer. So the stoichiometric ratio here is 1:1 molar ratio. But here there is one molecule of berberine chloride, and there are two molecules of amodine. So the stoichiometric ratio here is 1:2. For every one molecule of berberine, there is two molecule of amodine in one particular unit cell. So these are actually different from salts, because in salts there is involvement of ionic bonds, which is a type of a covalent bond, and these are also different from polymorph. We cannot categorize this as polymorph or solvates or hydrates. So co-crystals, it's a completely different class of compounds. And the advantages, a few advantages are, they have a good aqueous solubility and a faster dissolution rate compared to crystalline materials. Though the solubility advantage might not be as good as amorphous ones, but they have a better storage stability compared to amorphous solids. So this one particular aspects make them very attractive for improving the solubility. Earlier, there was only one option, that is preparing an amorphous. But now we can also go for the co-crystals, which also have good storage stability. And like as I said, the handling of amorphous, it's very difficult because of its hygroscopic nature and all. So, but here in case of co-crystals, there is no such limitations. They also have good mechanical properties like the compressibility, and also they have a good powder flow property. As you might have seen, materials that are hygroscopic, they usually tend to show a limited powder flowability. Common example that I can give is salt, sodium chloride, which we use. Suppose everyone might have noticed in rainy season that it doesn't flow properly. It tends to stick with each other or to the walls. So that makes amorphous materials also to have poor powder flow properties. But in case of co-crystals, these kind of disadvantages are not there. We can engineer them. We can prepare co-crystals like with properties that we need. And there are also been certain cases where the permeability has also increased of drugs using certain specific coformers for a given API. There was another question regarding the Ostwald's step rule. Basically, Ostwald's step rule, it states that suppose there is one compound while crystallizing, I'll try to explain it in some layman terms. So you're trying to take down the stairs. You directly don't go from the starting to the bottom. So you gradually step down, right? Similarly, while we are preparing a compound, if we consider these states as the energy level, so the first stair where the stair is starting, it's at highest energy level, and the lower one, it is having a lower energy. So if the end goal is to reach the lower energy state, you cannot directly jump from higher to the lower energy state. You need to take one small steps, and then you reach the lower energy state. So what I mean to say is that it's not the most stable phase. The most stable phase has the lowest energy. So it is not the most stable phase that is formed directly. Instead, when we are doing crystallization, maybe first amorphous form is formed, and then the next least stable crystalline phase, and then the least stable crystalline phase, and then the next phase, and then finally we'll get the most stable phase. So that's the Ostwald step rule, because the crystallization happens in a stepwise manner. It doesn't directly go from the solution state to the most stable state. So basically, that's the Ostwald step rule. And here the phase transition, the stepwise transition happens in by two mechanisms. That is by solution mediated and then the solid state transition. So the solution mediated, there is a solvent which can assist for the transition of the phase. So this happens very faster, and this is the most common type of phase transition that is seen. And there is another one which is the solid state transition. This happens usually very slow because there is no solvent to fasten the process. And this usually solid state transition can be fastened when the solute is in molten state. If we melt it, the phase transition can happen very faster compared to just solid sitting there. Like I said, amorphous, if we store it maybe for 6 months, then it will convert to crystalline phase. But if we try to melt it, it gives some extra energy. This energy is then utilized to stabilize the molecule. So that will happen maybe within 1 month. If we keep it in melt form, the phase transition can happen within a month or so. So this is the difference, like what we see in solid and then in the molten form. So these were the ones which I had got till yesterday. Maybe today also we have got a few queries. And if there is something you all need to ask, you can please share your questions. You can please ask the questions.
Yes, sir. Very good evening, sir. Sir, actually, I have a doubt, sir, regarding, metastable zone width. Sir, how can we design a crystal shape in the metastable zone width?
Okay. I'll do that. Just a second. Let me open the metastable zone width diagram.
Sure.
Yeah. So the phase diagram is visible of the metastable zone width.
Yes, sir.
Yeah. So what exactly was your question? Can you please repeat again? I'm sorry.
Sir, actually, I am asking how can we define the crystal shape or size during the metastable wedge
Okay,
zone?
okay. So metastable zone width, using this, we cannot exactly define the shape that we are gonna get because it depends on the solute solvent interactions that gonna happen. But what we can decide here is the kind of nucleus that are formed, and maybe we can predict the size of the nuclei that are being formed. Because if the nuclei size, which is very smaller, and there are large number of nuclei that are formed, then definitely we are going to get the final crystal size will be very small. So if suppose this is the metastable zone width, the region between the two lines. Okay, so on the right side it is sorry, on the right side it is unsaturated zone, means the saturation solubility has not reached in this particular phase. Okay, and in the left side it is supersaturated, it's a supersaturated zone. This region, it's very labile because here if we tend to carry out the crystallization, or if we maintain our uh, solubility or temperature at this particular point, there is a high chance of primary nucleation. So primary homogeneous nucleation. Primary homogeneous nucleation means the spontaneously nuclei are going to appear, and when they appear spontaneously, because of very high supersaturation, a very large number of small size nuclei are formed. Okay, so there is a lot of supersaturation which is being consumed for the formation of this particular type of nuclei, and it happens very faster and in an uncontrolled manner, because here whenever this kind of happens, we cannot control the rate at which the nuclei are formed. So when a large number of such small size nuclei are formed, maybe 50 to 60% of the supersaturation, which is the driving force for crystallization, it is consumed for formation of nuclei, and now there is no supersaturation or very limited supersaturation which is left for the crystals to grow. So as a result, what happens? The crystal size that we get at the end of the crystallization process, if we start at the supersaturated zone, the particle size will be very low. Generally, this kind of behavior is seen, and also the size distribution will be very wide, because there might be a few crystals which might have grown to a little bit bigger extent, larger extent, and then there might be few crystals which might have not grown at all. They might be like, you can get a size distribution anywhere between 50 micrometer D10, and then D90 it will be like maybe 700 800 microns. So this is a very wide size distribution, which is not basically um, it is not required for pharmaceutical operations, because as the size distribution is narrowed down, the predictability or its properties can be predictable. But when we carry out this kind of crystallization in the supersaturated zone, it tends to be very unpredictable in nature. So what we can understand from this phase diagram of the metastable zone width is, we always tend to perform the crystallization in this metastable zone, because in this zone the supersaturation, it's maintained at an optimum level, so that the level of supersaturation which we keep increasing over time, because when crystallization is going to happen, that means if crystal growth is happening, that means we are increasing the supersaturation, right, by decreasing the temperature, or maybe by adding anti-solvent or any such thing, or by solvent evaporation. So if we maintain in this zone, the amount of supersaturation that is being induced, it is in a controlled manner. So by this way, we can like ensure that the crystals, that whatever we are getting, it will be of narrow size distribution, and then it will not be of too small size, because small crystals, it can um, it will lead to various other disadvantages, like if the surface area is very high, the interparticular interactions will be very high, and this will lead to poor powder flow properties. So these are the aspects that we try to avoid during crystallization. So hope I was able to answer the question.
Yes, sir. Thank you.
Okay. So I'll take up the questions which I got from the Google forum. Or are there any other questions from those who have joined the meeting?
Sir, can you look at the chat?
Which chart? Chat. Okay. I'm sorry. What are the practical procedures to measure the induction time? Yeah, practical procedure that we have to develop basically, but I can give you a rough procedure which can be followed. We can use turbidimeters for this purpose. So basically, nucleation is the formation of some particular foreign or solid substances. Initially, it will be completely clear. The solution will be very clear. So there will be no turbidity, right? So when the nucleation starts happening, it will induce some turbidity in the media. So this turbidity can be measured. So the point where the turbidity starts appearing in the readings, that we can consider as the induction time, because that particular time is where the nucleus has started to appear, and there will be a sudden spike in the turbidity readings. So this is how we can say that the the induction has the nucleation induction has happened. Practical procedure exactly for these analytical methods, we have to actually develop our own methods in the lab, because it varies for different molecules or in different instruments. And yeah, so hope I was able to answer the question. Niranj.
Yes, sir. Thank you, sir.
So there was one question by Dr. Govinda Varadhi. What is the use of pharmaceutical crystal engineering in chemistry? So Dr. Govind, I guess, is not in the session. Anyways, I'm going to answer the question because it has appeared. So crystal engineering as such, for crystal engineering and chemistry, these two are like related fields. There is chemistry in crystal engineering, and also there is crystal engineering that can be used for in the chemistry field. So basically, I would say for chemistry, we can apply these principles of crystal engineering for the purpose of like for isolation of compounds, or for isolation and purification of compounds after synthesis, or for optimization of the yield that we are going to get after a particular synthesis, after the synthesis of a particular compound, or for maybe for the separation of enantiomers or diastereomers based on the solubility difference, if there is any difference. And also, if there are isomers that are formed, there will be differential solubility of a few isomers. Based on that, we can separate. So basically here, the most soluble one, we can use one particular solvent for dissolving one isomer, in which the other isomer is insoluble. So in this way, we can separate these two compounds. So basically, these are the broad areas where the crystal engineering can be applied. I got one message from Mr. Official. So can you please brief about PAT tools in crystal engineering? Yeah, sure. There is one whole unit dedicated for PAT tools. I guess unit 10 or something. I don't remember exactly, but yeah, I'll just give a gist of what PAT tools can be used in crystal engineering. So as I said, one, the turbidity meter, that's also one kind of a PAT tool which can be used, which is used for the study of the nucleation induction time, to actually monitor the in the real time when the nucleation is starting, or when crystallization is getting completed. Suppose, how can we know when the crystallization is getting completed? We have one particular, suppose after the formation of nuclei, there was initially zero turbidity, and then it went to 5, maybe to the 5 points, and gradually it started increasing, and it started maintaining at a point of 7 or something. So, or it was increasing. We can get a curve like this. So the point where crystallization stops, the turbidity measurement also will stop increasing. So that point you can consider as the point where the crystallization is completed. So, and then we have like the particle vision measurement, and then the FBRM, focused beam reflectance microscopy, which they actually gave idea about how a particle is growing in a real time, how the size is increasing of a particle, or how the shape is being developed for a particle. So we can use these probes, and then we can monitor their size measurement at the time of nucleation, or we can also measure what was the nucleus size initially which formed, and till what size the nuclei grow. So these kind of measurements can be done with FBRM and PVM. And also we have Raman probes and all, which uh, Raman probes particularly they have been used for this anti-solvent crystallization to check the level of anti-solvent and solvent in real time. We know we are adding, but there can be different probes placed at the different locations, and then we can see how efficient is the mixing process, or also for drying, for the real-time monitoring of the solvent levels in our crystal samples. So these are the few PAT tools which are being extensively used or for in crystal engineering. So in detail, there will be one more chapter in chapter 10. That time maybe we can get into more details about these PAT tools. Yeah. So can I move on to the next question, please? Okay. Can I just take up these questions and then I'll come back to the classical nucleation theory, please?
Sure, sir.
Okay. So the next one is can we apply pharmaceutical crystal engineering on formulation of cosmeceuticals? If yes, how? Yeah, surely this pharmaceutical crystal engineering, it can be applied for cosmeceuticals. Suppose there are a few vitamins, basically vitamins. I'm taking a general example because they are most commonly used for cosmetic purposes. So most of the vitamins, they are light sensitive, or they tend to undergo oxidation. So maybe we can use our crystal engineering principles, like prepare co-crystals for the co-crystals, and then try to reduce their light sensitivity, or its uh, we can maybe prepare co-crystal with antioxidant so that it itself does not undergo oxidation. Or maybe we can use it as for synergistic purposes, like we can combine two materials, one which is having maybe anti-aging property by some other mechanism, and another antioxidant will co-crystallize with that particular compound, and then its activity will be increased because now it will give both anti-aging and antioxidant properties. So these are a few ways uh, by which crystal engineering can be used for improving the efficacy of cosmeceuticals, basically. And also we can like do the polymorph screening, because a few polymorphs, we know key polymorphs, they tend to have show different activities, or one of the polymorph can be active and another polymorph can be inactive. So we can know this which polymorph is active, and then using the crystal engineering principles, we can isolate, or we can get only the active polymorph type. So these are the kind of applications which can be, yeah, done for cosmeceuticals. So next question is, how can we design a crystal size during metastable zone width? I guess I answered this question. Yeah. So another question by Harshal Srivastava. Can you explain how secondary crystallization is impacted by supersaturation levels, and what are the implications for crystal growth and nucleation rate? Yeah, as I said already, at high supersaturation levels, uh, usually a large number of smaller size nuclei is formed. So this means that the final size distribution that we are going to get, it will be at a smaller size and also a very broad one, because it's uncontrolled. So in case we are doing seeding crystallization, because secondary nucleation, it's like basically seeding crystallization. Suppose at the time of introduction of the seeds, the supersaturation level is very high. So at that time, what happens? already there is a surface which is conducive for nucleus to form on that particular surface, because as we have seen in the mechanism of secondary nucleation, there is one surface which improves or which increases the rate of nuclear formation on its surface because it provides, because it lowers the energy barrier basically. So now the energy barrier is also reduced, and also there is high supersaturation. So now what it will happen, the crystals whatever we have introduced as seeds, that will be having a particular size distribution, and those also will grow. And on top of that, on these crystals there will be formation of very large number of nuclei. So I guess there was one mechanism called as the polycrystalline or needle breeding, I guess, polycrystalline breeding. So in that mechanism, what happens at a higher supersaturation level, the type of crystals that are usually formed, they are fibrous. They resemble fibrous morphology, or they resemble needle-like shape. And yeah, needle-like shape, it's usually not preferred unless that's a habit that we are gonna get after using every combination of like parameters. Unless and if we can avoid these needles, well and good. If it is not avoidable, then yeah, maybe we need to go with needles. But what happens is a API which can crystallize as plates or maybe cubes, but if we perform crystallization at higher supersaturation level, that will again form needles instead of cubes. So usually we tend to avoid these kind of things by maintaining the supersaturation level low, like the carrying out the crystallization in the metastable zone bed, where the supersaturation level is not that high. It is just enough to what do you say, to feed the crystals with sufficient solute, so that it doesn't form primary nucleus of its own. It will just grow the already formed nuclei. So in this way, we can control the particle size distribution and then also the shape. So as a general rule, whatever any crystallization that happens very in a faster duration, at a faster time, it will tend to form needle-like crystals. That is the general observation that we have seen, even in the lab or in the literature. So if the supersaturation ratio is very high, or the supersaturation is induced very faster, it will usually lead to formation of needle-shaped crystals or crystals with very high aspect ratio. So I shall hope I was able to answer your question. I guess Mr. Harshal is in the meeting. Can you please confirm once, or should I add on? So I'll can you please tell which part actually I need to explain? Like
Hi, Madhu. Am I audible?
yes, Mr. Harshal, you're audible.
Yeah, one question given for regarding the secondary nucleation.
Yes.
So secondary nucleation, how is impacting on the crystal growth and the size of the crystal? So when the
Yes.
supersaturation is shifting to the like what is the
Higher
design
levels.
higher level? So how is the
Yes.
impact that? And what will be the
Okay.
content?
There is one particular mechanism of secondary nuclear formation, that is by polycrystalline breeding. What happens in this case of polycrystalline breeding is that or needle breeding, we can also call it as a needle breeding. Just share the slide. Just give me a second, please. So is my slide visible? So yeah, so as we can see in the second mechanism of the secondary nucleation theory, which is the needle or polycrystalline embedding. What happens here is at high supersaturation level, we usually tend to get needles or dendritic crystals because these are the easiest crystals that can be formed compared to any other type. So the disadvantages of this is that they tend to undergo fragmentation. And suppose a needle which is having size of initially maybe 200 micrometer, now it tends to break up. Okay. So after it breaks down, maybe we'll get multiple particles which have size ranging from maybe 2200 micrometers. So what this does is this will basically impact the particle size distribution first of all. So now we tend to have particle size distribution which is very broad, because now we are getting particles from 20 micron, which are the fragments, and also 200 micron or 100 micron, which is the initial needle, because not all the needles tend to break. A few might break down, and a few. I'm sorry, there was some problem. I don't know what happened. The application just crashed. Yeah. So I was explaining that at high supersaturation level, what happens is that needles or dendritic kind of crystals tend to form at a very faster rate. So this we usually we do not need, because as I said, they are susceptible for fragmentation, and when they undergo fragmentation, we usually get crystals which are having very wide size distribution, and very wide size distribution. That means the flow property is not good, or maybe the compressibility of the crystals might be compromised. So these are the problems that we get. So in order to avoid that, higher supersaturation beat any kind of crystallization. We never initiate crystallization process when the supersaturation is very high. So Mr. Harshal, any further clarification needed for this?
No, thank you.
Okay. Okay. I'm sorry for the disturbance. Yeah. So there was a question from Mr. Abhay regarding the classical nucleation theory. So Abhay, can you please confirm if I have to explain the complete classical nucleation theory, or there are some parts which you might have not understood that I might have to again re-explain? You can drop in a text.
Actually, sir, can you please re-explain the theory?
Hello, Abhay, are you there?
Yes, sir, I am here.
Yeah.
Sir,
So I was
actually,
asking if I need to explain the whole classical nucleation theory, or you just need a part of it, like where I have a few doubts, like particular doubts.
sir, I just need a brief introduction regarding theory. Sir, rest of sir derivation I will understand.
Okay. So yeah, the classical nucleation theory, basically it was postulated, it was theorized to explain how crystals are formed from how crystallization actually takes place. So the CNT was actually developed by Weber and Vollmer, and they actually developed that from by considering the example of condensation of a vapor into a liquid. Okay, so this was not taken, or this was not explained in terms of solid crystals forming from solution phase, but rather it was, they considered the example of vapor. Suppose you collect a vapor somewhere and you condense it and collect, it will form a liquid droplet. So this was the mechanism that they considered to explain the how nucleus or the nuclear clusters are formed. So I'll share the screen as well, so that will be easier for you also to follow. So yeah. So as per this particular process, they say that the nucleation process is actually competition between the supersaturation. As we know, the supersaturation is the driving force for the formation of crystals. So it is the competition between the supersaturation, the driving force, and the surface free energy, which resists the formation of crystals. So there is one thing which wants the crystal to form, which is the supersaturation, and there is another thing which is the free energy, which wants the crystal to not form. So this is a competition between these two factors. So whatever takes an upper hand, that particular process will decide whether crystals should form or not. So our motive whenever we are doing crystallization, whenever we are performing crystallization, is to maintain a higher supersaturation level, so that the driving force is always above the resisting force. Okay, so the phase transform. So in this case, when we maintain the higher supersaturation levels, always there is a phase transformation which happens. Phase transformation may be from the molecular form to the solid form, like the formation of a nuclei. That is a kind of phase transformation, because earlier there will be a homogeneous solution, clear solution, which is completely in liquid phase. So from liquid phase, we'll start getting solid crystals. So this is a phase transformation, kind of a phase transformation that happens, and it involves the reduction of the free energy. As free energy is reduced, I'm really sorry, I don't know what the issue is, but yeah, the Zoom is crashing a lot. Yeah, continuing with the classical nucleation theory. So yeah, so when the actually the phase transformation happens when there is a reduction in the free energy. So that means whenever the crystallization process takes place, the free energy reduces. As we know, the energy in solids is lesser than liquids, which is in turn lesser than the gases. So when the phase is transforming from, or when new phase is forming from liquid to a solid phase, the energy is basically reducing. So the free energy reduces and then the crystallization happens. So as per this theory, it's a one-step process where the clusters, they aggregate spontaneously. It's like they get generated automatically from a liquid phase to form a nucleus, and it proceeds through the addition of monomers until a critical size is reached. Suppose, just imagine there is a room, an empty room, and then suddenly a few molecules start appearing here and there. And because these molecules are they resemble each other, there might be some kind of force that might be trying to keep them together. So what they will do, whenever such kind of molecules are formed here and there, they try to come and form a agglomerate kind of thing, I can say. Okay, so this kind of aggregates which are formed, this further proceeds. I mean that it grows by the formation of monomers. Suppose there were 10 molecules that were formed initially at 1 second. Okay, they form an aggregate, and then at 15 seconds there are 2 to 3 more molecules which are formed here and there. These monomers, this we can consider as monomer. Each molecule, this comes and joins the aggregate, and another molecule which is another monomer which is formed, it will also come and join this particular aggregate. So this joining process, it will continue till a critical size is reached. So what is a critical size? Critical size is something which above which the crystal growth happens, or a size which we can call, it's like a cutoff size for calling a particular cluster as a nuclei, because right now we are calling them as clusters, right? So in order to call these clusters as nuclei, it has to reach one particular size, critical size. So that is where we will start calling them as nuclei, right? So this proceeds until the critical size is reached, and then whatever nuclei or whatever clusters which are smaller than this critical size, which is also called as a critical nuclei, they again dissolve back and dissolve back in solution. And while those clusters which can grow above this size, particular critical size, they proceed with crystal growth phase. As we know, crystallization process, there are three phases. One is crystal nucleation. After nucleation, the crystal growth phase takes place, and then the termination phase. So only those nuclei which are formed in the first phase, which are having a critical nuclei size, they proceed to the second phase. Otherwise, again, they will redissolve back, and they will contribute to the formation of other nuclei which are already formed, or the other clusters which have outgrown the critical nuclei size. So, and it also explains this particular theory that the cluster size is inversely proportional to the level of supersaturation. Higher the level of supersaturation, smaller the cluster size. As I already told, if we maintain higher supersaturation ratio, the amount of the size of the nuclei that is formed will be very small, right? And also there will be numerous nuclei that are formed compared to the nuclei that are formed at a lower supersaturation levels. So the cluster size is inversely proportional to the level of supersaturation. Higher the supersaturation, smaller the size of the clusters. And as the saturation increases, the supersaturation ratio, if we keep increasing that, the critical size for the nuclei also decreases. That means, suppose if we consider two supersaturation ratios, one is 1.5 and another one is 3. Supersaturation ratio of 1.5 and 3. Suppose the critical nuclei size for at supersaturation level 1.5 will be maybe 5 micron. Okay, but in case of the supersaturation ratio of 3, the critical nuclear size will be maybe 1 micron. So as we increase the supersaturation ratio, the critical size that is required, which will further proceed for the crystal growth phase, it will decrease. So higher the supersaturation level, lower the crystal size, and the critical size for the nucleation decreases, and the crystallizing solute is more likely to form a nucleus. So when we increase the supersaturation ratio, nucleus will get at more numbers, and it is certain that we are going to get some nucleus. And compared to lower supersaturation ratio, there might be probabilities that the nuclei won't form at all, because the kind of barrier, the energy barrier that has to be breached for formation of the nucleus, that might not be sufficient. So supersaturation is the driving force, as I said earlier, and that is gonna contribute for the formation of nuclei. So
Okay, sir. Thank you. I was talking about only first slide.
okay, then no problem.
Thank you, sir.
Welcome, Abhay. Are there any other questions? Regarding the link for the article that I had shared earlier, that I had showed earlier. I'm just gonna drop. Just give me a second, please. Yeah, I have shared the link to the article. Is there any other question from first week that I can take right now? I can answer. Okay. And in case anyone has any questions, yeah, feel free to drop a mail in the forum. There's a discussion forum which is open. Yeah, you can always drop in a text for that, or drop in a mail. We'll reply as soon as possible. Okay, there was one more question which we had got today 4 hours ago at 1. So it was tell more about co-crystallization of drugs and their methods. I'll just tell in brief, because I guess week 6 is completely about co-crystallization. So yeah, co-crystals, basically these are prepared from by using API and a coformer, and usually the type of interactions that are involved here are weak hydrogen bonds or the van der Waals interaction, pi interactions, etc. So this, basically we are using co-crystallization method for predominantly to improve the solubility of compounds, because there are many BCS class 2 drugs which have BCS class 2 and 4 drugs which have very poor solubility, which again impacts its bioavailability. So we are gonna use this co-crystallization method to improve the solubility and enhance the bioavailability, and maybe there might be a few compounds which are unstable as such. So we are gonna co-crystallize them with another coformer which is having a better stability to improve the stability of the overall molecule. So the stability might be increased by co-crystallization, or we can also improve the permeability, like BCS class 3 drugs. It is having excellent water solubility, but there is a limited permeability of that particular molecule. So for also improving the permeability of co-crystals, we can do of APIs, we can perform the co-crystallization. And these were the few applications. And what are the methods we usually follow, like wet grinding method, dry grinding method, or slurry based preparation method. We can just dissolve API and coformer and then evaporate the solvent, or we can mill API and the coformer in presence of a little amount of solvent, which enhances the hydrogen bond formation or the interactions between the API and the coformer. So these are the widely used methods that are followed for co-crystal preparation. In detail about what kind of coformer we need to select, what are the parameters or the selection criteria, what rules are there. There are many rules, like the pH rule is there, pKa rule is there, pKa rule is there, and then the hydrogen bond acceptor donor, and then there are a few computational tools which we can use to predict what kind of coformer will be is fitting for one particular API based on the interactions, based on the electrostatic potential surfaces that is it's going to generate. So these are a few tools that we are going to cover in the week 6. Are there any other questions from maybe others? If there are no questions, maybe we can conclude this particular interactive session.
No, sir. Thank you.
Thank you. Bye. So thank you everyone for attending this particular session. And yeah, in case anytime you have any doubt, yeah, or any clarification is needed, just drop in a mail in the Google form, or you can just reach us out. And yeah, happy learning. Thank you. You guys can please leave the session. I'll